Hello, in this video, we're going to be talking about the lysosomes, the cytoplasm, the cilia, and the peroxisomes. So let's not waste any of your time. Let's get right to it. Alright, so again, we got four in this one. Lesser known, not lesser known, but lesser detailed. So we got four in one, starting with the lysosomes. Now, when we're talking lysosomes, which you've already mentioned, part of the endomembrane system, we've talked about the engulfing of material, and with the digestive enzymes on the inside, designed to break those down and to get those parts out. Now, when we talk about when we get to chapter 5 and we're looking at the plasma membrane, we're going to be looking at systems, kind of bulk transport systems, and we're going to showcase how what that process looks like when a lysosome or a vesicle has to attach with the membrane to release materials out into essentially the bloodstream, to get rid of it so it can be removed and gotten rid of. We'll look at that in chapter 5. Now, lysosomes are made in the Golgi apparatus. Okay, and again, they've got those digestive enzymes, the idea of breaking down macromolecules or molecules of any kind is their function. They also play a part in the immune system by, one of the things they may engulf is foreign material. It could be a virus, it could be bacteria, it could be bacteria that has been destroyed or broken apart by antibiotics, and then therefore the lysosomes come along and engulf those and get those things broken down. And when it does engulf something, okay, I mean, it literally touches it and then pulls it into itself, essentially. It'd almost be like if you were thinking about eating the way a lysosome does. Instead of, say you had a chicken leg, instead of putting it, taking a bite and chewing it up and swallowing it down your esophagus into your stomach, you would take the chicken leg and just kind of push it into the skin on the outside of the stomach, and the stomach would then, or the skin then, would wrap around the chicken leg and pull that into the stomach, and that's how it would get into the stomach if it did it the same way that a lysosome does it. Okay, but here is this is the macrophage. Macrophage is part of the immune system, and this is how it incorporates lysosomes, or how it does its job by using lysosomes. You can see in the picture that the macrophage kind of has an opening, and that foreign material has entered the macrophage, and then the lysosome comes along and engulfs that foreign material, and then in essence breaks it apart, and then you can see it goes to the exit area and just releases it out. But that's the idea of how the immune system can assist or help the immune system in that manner. All right, so this is our fourth member, or our last member to talk about, of the endomembrane systems. We just want to remind you about this elusive group and then what they do, what each member does. Remember, they're the protein-making organelles. At least they deal with proteins. Ribosomes make them. Raphiar modifies them. Golgi packages them up. And in some manners, in the vesicles, which kind of lysosomes are lumped with, are the trucks, and the proteins get in there and they're transported. But then lysosomes also can break them down as they're old and worn out. Okay, our next organelle to talk about, peroxisomes. Peroxisomes are pretty important. They're certainly lesser known. If someone was to list off organelles, this is one they may not mention. It doesn't get a lot of press. But they're, as you can see, kind of a small, round organelles. They do have a membrane, as they all do. But they got pretty important jobs. They're breaking down fatty acids and amino acids. So again, they're just around the cell, and when they come in contact with these, they help to break them down. And they get their name because through their breakdown process, a product that comes out of that is hydrogen peroxide that gets released. And so therefore that's where they get their name, peroxisomes, peroxide. But not only breaking down fatty acids, amino acids, they can also detoxify poisons like alcohol. So in an organ like a liver that has that task, you'll find lots of these. So it isn't necessarily the liver that's doing that. It's all these peroxisomes in the liver or whatever the organ may be that's breaking down stuff. Okay, this next organelle we've already mentioned during the prokaryotic, eukaryotic talks. We talked about the flagella, and it's super important organelle for motion, long hair-like structure, and as I mentioned in the previous video, you can think boat motor. When it's whipping around, where's my hand at? There it is. When it's whipping around, it can propel, in this case, the bacteria into a particular direction. But if you want to think transportation, think boat motor, one large powerful motor moving the boat in a particular direction. Our next organelle to talk about is the cilia. Cilia, again, used for motion. Okay, but when you think about this as far as motion, because they have many, they have lots of them, as you can see there in that picture beside me. Think about the movies that you've seen with those big Viking ships as they're going across the ocean, and they've got 10, 15, 20 people on each side with those long oars rowing in unison. You got the guy in the front or back going row, row, to keep that rhythm going. That's like cilia. Lots of them. Just kind of moving, helping to move us around. But again, short, hair-like structures as opposed to the flagella, which is long. So you also have another role when they're inside of systems or organs like your nostril, okay, or the intestines. They can help move products. So like your nose hairs or cilia in your nostrils can help to trap dust. If it's in your intestines, it can help to move food along the process. That is cilia. Okay, one thing we want to think about, make sure that we understand, is the differences between the flagella and the cilia. Now, they're both microfilaments. Microfilaments may not be a word that you're familiar with, but that is an upcoming organelle that we're going to talk about. But they are microfilaments, so they're the same structure, but just have different arrangements. Obviously, the height alone, the length alone, will tell you that. So they're made from the same thing, but different structures. Flagella are long, okay, and there can just be one, polar, or as we saw the Perotrichus, there can be many on both ends, one on both ends, one on either end, a bunch on one end, bunch on both ends. Pretty variation, lots of variations there. But cilia is short, and they are many. But again, microfilaments, just different structures One's long, flagella. One's short, cilia. Can be just one. Excuse me, or there can be many. Well, that's our video over the lysosomes, flagella, peroxisomes, and cilia. Next time we get together, we'll be looking at number 8, the cytoskeleton and the cytoplasm. So we'll see you then.